So our first plenary speaker is Professor Stefan Borgwardt, who is the head of the Neuropsychiatry and Brain Imaging Research Group at the University of Basel in Switzerland. He is a clinical psychiatrist and a neuroscientist at the Department of Psychiatry, also at the same institution. He's also a visiting professor at the Institute of Psychiatry in London, at King's College in London. So, and the title of his talk today is Third Generation Neuroimaging, so translating the research evidence into clinical utility.
Hello, everybody. Thanks a lot for the very kind invitation and the few words and introduction. My talk will be about neurobiological correlates, neurobiological findings in early psychosis, and in particular, I want to focus on neuroimaging. And as the title suggests, I will finish my talk with the so-called third generation imaging, and I will go through from first generation imaging, from the beginning of psychiatric imaging, via second generation imaging and third generation imaging. And the idea is to give you an idea why we do neuroimaging now and what the potential clinical applications of neuroimaging methods are in future. So what is clinical imaging now? It's rather simple. We use neuroimaging as psychiatrists purely to exclude organic diseases like brain tumors or any clear neurological diseases that may explain psychiatric symptoms. And this was first done in the 70s with CT, computerized tomography, and is nowadays done with MRI. And yeah, whatever you are interested, we can look at gray matter, white matter, CSF, and but the main reason really is to exclude any organic findings. So the most replicated clinical relevant finding in schizophrenic patients is a ventricular enlargement. This has been replicated for numerous times, and the first time this was published was in the 70s by Eve Johnston. In the last, I'd say, 10 to 20 years, there was a shift from these rather simple clinical applications towards more sophisticated questions and techniques. So in the first step, structural brain alterations in the frontal lobe or wherever were associated with some kind of disease. And nowadays, with the more and more coming neuroscientific methods, whole brain automated methods as voxel-based morphometry, white matter methods as tractography, where you can follow certain tracts within the brain, and functional brain imaging allow to see how the brain works when it's alive. And apart from that, there are other advanced neurochemical and pharmacological imaging methods as PET and MRS. I won't go into detail, but that you get an impression that there was a big shift from the simple clinical imaging towards these methods. And in the last time, there were multi-center image analysis studies and multimodal imaging studies. Multimodal in that sense means that there are combinations of structural and functional imaging studies. I will now give you an overview of structural and functional imaging findings in early psychosis using these methods. But why actually do we do neuroimaging in psychiatry? What's the rationale behind? And in 2006, Andreas Meyer-Lindenberg and Daniel Weinberger, they proposed, or they gave a rationale for applying neuroimaging methods that may kind of bridge the gap between the genetic basis of some brain diseases and the actual symptoms or syndromes. For instance, as you see here, schizophrenia is related to severe working memory deficits, and these working memory deficits are associated with functional abnormalities in the prefrontal cortex, and these are related to certain genes. However, up to now, there are no consistent or reliable anatomical or functional alterations that have been unequivocally associated with psychosis or schizophrenia, and again, no clinical applications have been developed in psychiatric neuroimaging. That's the case now. Pat McGorry already showed the upper panel of this slide. I want to shortly mention the staging model of schizophrenia. And stage 3 refers to acute psychosis, or we could say first episode psychosis. I'm sure you will see some results in the next talks about that. I will mainly focus on stage 2 and stage 1, the so-called prodromal states of psychosis. And again, you have seen this picture before, and what it shows is that actually 2 to 5 years before the actual onset of psychosis, on the right side, there is a prodromal phase with so-called attenuated psychotic symptoms, which means you have not severe, but attenuated psychotic symptoms. So you're not delusional, but you're not sure if the world around you is as it was before, or you have hallucinations, but not as often as you would have in a psychotic episode. And another inclusion criteria or characteristic of this prodromal high-risk group are patients who have very brief, limited, intermittent psychotic symptoms, which means that the symptoms disappear without any treatment within a week of time. And all the figures you will see in the next few minutes refer to these ultra-high-risk patients. And as mentioned before by Pat McGorry and Ashok Malla, the overall aim of this kind of research is to either prevent the development of psychosis or to develop treatments. A meta-analysis published in 2012 showed that roughly 35% of all patients labeled as ultra-high risk will finally develop psychosis. And you might now think 35% is not very high, but as discussed before, the other 65% of patients are not fine, they are not healthy, but they will not develop psychosis. One reason that they are unwell is that they not only suffer from psychotic symptoms, they also suffer from severe cognitive abnormalities. And if you compare all the high-risk patients or high-risk individuals published so far with controls in a meta-analysis, you see that they have an overall relative deficit in cognitive functions. And if you further compare those high-risk individuals with later transition compared to those without, you see an even further relative decline of certain cognitive functions. So apart from the psychotic symptoms and symptomatology, there are other symptoms as cognitive functions, as social functioning, which are disturbed in this high-risk group. The first big study that has been published on grey matter deficits in high-risk individuals was published in 2003 in The Lancet. Christos Pantelis and his group, they showed that patients had severe gray matter reductions in the parahippocampal lobe, parahippocampal gyrus, and in the hippocampus, and in the orbitofrontal cortex 1 to 3 years before the patients developed psychosis. And interestingly, these gray matter deficits, they further declined, or the gray matter further declined via the transition to psychosis. So patients were scanned at 2 times before the actual development of psychosis and afterwards. And in Basel, we more or less replicated the study with surprisingly similar results. As I said, I want not only show you the results of single studies, I also want to show you multimodal studies, multi-center studies. And one multi-center study here, again a voxel-based morphometry study looking at gray matter volumes in patients at high risk for psychosis, had included patients from Melbourne, London, from Munich and from Basel. And again, they showed gray matter deficits in patients at high risk of psychosis. And there were another cohorts of high-risk patients. I mainly spoke about this so-called clinical high-risk cohort, which means that these patients have some kind of psychotic symptoms. These psychotic symptoms are attenuated. They are not as long as other psychotic symptoms. But there is another high-risk group, and this high-risk group is purely based on a genetic high risk. So people with a first-degree relative of psychosis have an increased risk of psychosis. And these patients are investigated in this Edinburgh high-risk study for, I think, 20 or more years now. And they showed a slow decrease of gray matter in the prefrontal cortex over time. And this decline was not only shown in the high-risk subject, it was also present in healthy controls, of course. From the age of 35, there is a physiological decrease of gray matter in the brain, but compared to these healthy controls, there was a significant difference to those who had a high risk and further developed psychosis. We've also done some voxel-based meta-analysis, now including about 1,000 high-risk subjects worldwide. The problem of all these high-risk studies is that because it's very, very difficult to find these patients, most centers have only 50 or 100 or maybe 200 included subjects. So all the studies that have been published so far are based on relatively small sample sizes. So it was important, or it is important, to have some kind of meta-analysis and to confirm the results. And again, these meta-analyses showed that there were reductions in the superior temporal gyrus, in the anterior cingulate, and in the hippocampus. And there were other regions within the temporal gyrus and inferior frontal gyrus which were specifically related to later development of psychosis. As you might have heard, many of the studies looking at structural abnormalities in schizophrenia patients and also in first episode psychosis patients are more or less confounded, or at least influenced by intake of medication, in particular of antipsychotic medication. And there was one study published in 2011 by Ho and Andreasen, and they showed that the more antipsychotic medication was given to a patient, the smaller the gray matter in the brain. And this was shown over a long-term period. And critics said, okay, that's clear, because the most severe patients get the highest dosages of antipsychotics, so this explains everything. But illness severity had relatively modest correlations with tissue volume reduction and drug misuse, and had no associations when effects were adjusted. So this indicated that antipsychotic use has at least an influence on all these imaging results in schizophrenia and psychosis. And therefore, apart from the clinical need of studying patients at high risk of psychosis, just for scientific reasons, it's very, very important to have this ultra-high risk group and unmedicated first episode group. Because we actually don't know if the gray matter reductions we've seen are only or partly related to antipsychotic use. So we did a further meta-analysis, but included only antipsychotic naive high-risk subjects and antipsychotic naive first episode patients, and looked for differences compared to controls. And again, you see gray matter reductions in the anterior cingulate and in the superior temporal gyrus compared to high-risk subjects. And you see, when you look at differences between controls and high risk, again, differences in the temporal lobe. Interestingly, these gray matter reductions were related to clinical symptoms. So in the superior temporal gyrus, the lower the gray matter, the higher the severity of psychotic symptoms. I'd like to emphasize that when we talk about neuroimaging methods and results, it's always necessary to have correlations or any relation to the clinical symptoms or clinical outcomes. It doesn't make sense, from my opinion, just to show nice images with differences here and differences there. It has to be shown that these differences have a clinical meaning. So, going a bit further from structural imaging now to functional imaging methods, and one particular method which is investigated by many groups is functional MRI, and this uses the so-called Bolt effect, which basically means that the oxygenated hemoglobin has different magnetic characteristics compared to deoxygenated hemoglobin. It basically means that if a particular region in the brain is has a high cerebral blood flow, it changes the signal in the MR scanner. So basically, you measure a change in cerebral blood flow, and you think, and there is a correlation to brain activity. But we have to be aware when we talk about brain activities in functional brain imaging that we actually speak about changes in cerebral blood flow, and this is only an indirect measure. So, putting together all published fMRI studies in patients at clinical high risk, again, they showed reduced activation in the inferior frontal gyrus, in the superior frontal gyrus, medial frontal gyrus, and the anterior cingulate. So now you have heard these regions, I think, third or fourth time, and it should show you that there are structural deficits in these regions, and there are also functional abnormalities in these regions. And one very interesting study was published by the group here from Montreal in the last year. I'm sure you will hear about the study later, but what I want to show here, I'm sure you can explain it much better than me, was that there were functional MRI abnormalities shown, and these were clearly related to clinical outcomes, in this case to remission or non-remission. And these are the kind of neuroimaging studies we and all the clinical doctors want to see. We have recently looked at connectivity abnormalities predating the onset of psychosis. Again, we looked at this high-risk group and were interested if medication had an effect on brain connectivity in first episode patients. And the first thing we saw that was the regional brain activation during working memory task was reduced in high-risk subjects compared to controls. And the first episode were, in general, they were also reduced. But if we compared, but if we had a look only to the non-medicated first episode patients, there was a contrast to the controls. So that could indicate that for the specific connectivity between the frontal gyrus and the superior parietal lobuli, there was a kind of normalizing effect of antipsychotics. And again, as I mentioned before, it's important to show that these neuroimaging findings have a relation or a meaningful relation to clinical symptoms. And here we show that abnormalities in working memory induced modulation of connectivity was able to separate healthy controls from people at high risk of psychosis. And you see here at the bottom the amount of hallucinations, and the higher, there was a clear correlation between the amount of hallucinations in the arms and in the non-medicated first episode patients. And one next step of putting together all these fancy findings is to integrate structural and functional brain abnormalities. And unfortunately, it's not that easy as I expected, at least. I thought that if you have structural deficits, so less gray matter volume in a certain region, then you should have less brain activation in this region. But this is not necessarily the case, as I show you later. Or you could put it the other way around. You could expect that if there is less gray matter in a region, then you have compensatory mechanisms that lead to increased hyperactivity. And therefore it was important to control the functional abnormalities for structural abnormalities. And one meta-analysis was very recently published, and it showed you areas in the brain which show decrease of gray matter volume related with hypoactivation. This is a pink color. So that's what you actually might have expect. Less gray matter, less activation. But on the other side, there are also regions with a decrease of gray matter volume with either hyperactivation or a failure of deactivation. And this indicates that interpretation of these kind of neuroimaging findings is not that easy. And finally, the right figure again shows differences between antipsychotic naive first episode patients and medicated. this suggests that antipsychotics may specifically target regions in the brain. So what might third generation neuroimaging mean, or what can we understand under translational imaging? It should definitely move away from the simple investigations of neurobiological correlations. It should actually translate imaging findings in the clinical field, targeting either longitudinal outcomes as transition, non-transition, or remission. We will hear about that in a few minutes. And what is very interesting as well, response to preventive interventions. And for this reason, we need to move away from group analysis to individualized neuroimaging. All the studies you have seen before were in groups of 20 versus 20 patients versus controls, or these kind of meta-analysis. But for the individual patient, and for us as doctors and therapists, it doesn't help to say, okay, you're a group of patients, or you belong to this group of patient, and this has a reduced gray matter volume in the prefrontal cortex by 20%. So we have to have techniques that allow an individualized diagnostic tool. And there are mainly few groups in the world who use these kinds of pattern recognition techniques. I'm not a particular expert in this, but what I understood is that instead of using an a priori hypothesis with an a priori region, you or the software looks for certain patterns across the whole brain. That means that gray matter reductions in certain regions are related to relative gray matter increases in other regions. And then you get a kind of pattern map for each group. And this tool will look for regions which contribute to the classification of certain groups. And in the end, you can put one single patient into this model, and he will classify this patient to a certain group. And then you can double check if this was correct or not. And Nikos Kotsouleris from Munich, he mainly did these studies. He published a first paper in 2009 in the archives in the Munich cohort, and this was highly discussed. And he replicated these findings with a different cohort, with the Basel cohort. And what it shows is that a simple 10 minutes MRI brain scan can, or with the help of this brain scan, you can predict with a likelihood of 80 to 85% if a high-risk patient will develop psychosis. Now you can remember to the 35% at the beginning of my talk. 35% of all high-risk patients will develop psychosis, and this is based on a clinical evaluation. And that's very important, within the ultra-high-risk cohort, we can further increase the prediction of psychosis to 80-85%. Of course, as I mentioned before, these studies are small. These included roughly 50 high-risk subjects, so we have to be cautious. But also in other neuropsychiatric conditions, as Alzheimer's disease, as Huntington's disease, as in multiple sclerosis, all these fields use these kind of methods to classify individual patients and to allow individual prediction of clinical outcomes. So to sum up, there are bilateral prefrontal limbic and temporoparietal cortex reductions, and these are correlates of an increased vulnerability to psychosis. And those high-risk subjects who will later become psychotic, they have further gray matter reductions in the superior temporal gyrus and inferior frontal gyrus. And we expect that these changes are crucial to the development of psychotic illness. Functional and structural abnormalities in the high-risk group may share a common pathophysiology, not in all regions. And prediction on outcomes and at single subject level might be a promising tool to translate imaging findings as clinical tool in future. So the work which has been presented here has been done in Basel and in London. I'd like to thank my colleagues and you for your patience, and thanks a lot.
Thank you, Stefan, for a great overview of brain imaging in early psychosis. I think it was very clear, very, fascinating results that you've presented. So we have time for maybe one or two questions. I believe you were supposed to write down your question, but I don't believe we have received any. So, but perhaps if you want to stand up, if you have any question, please do. Yes. Yeah. Could you use the microphone, maybe? Yeah, just to repeat the question, so your question was about what kind of MRI protocol was used as part of those different studies presented here. Yeah.
If you refer to the last slide, this was done with a relatively simple three-dimensional scan, which took 10 minutes time. It's not a regular clinical scan. It's not a simple T1 scan. You need 3 dimensions, otherwise it doesn't work, because it looks for 1 millimeter voxel within the brain. But it's not like a functional brain MRI scan where what takes an hour or so, and which is very time consuming and demanding. You need some facilities. You can apply this sequence whenever you have an MRI scanner. That was only one scan, and there was a clinical follow-up period of 2 years, a minimum clinical follow-up period. But this was just one scan.
We'll take a last question.
Do you have any evidence between the population who converted, who translated in affective psychosis, or those who translated in schizophrenia type of psychosis?
For the, I can't tell you for all the studies. I mean, in the, I think for the general high-risk population, it's about roughly 50 to 60% who will develop schizophrenic psychotic episode. In Basel, we had a slightly higher proportion of people who finally developed psychosis. I think it also depends a little bit on the inclusion criteria. Also, the inclusion criteria across these worldwide early detection centers are very similar. There are always some differences. So, for instance, the German inclusion criteria, they, I can show you, they also include so-called basic symptoms, which are self-recognized disturbances of concentration and so on. So they have a slightly different risk. And yeah, does this answer your question?
So if there is specific voxel-based data or functional imaging data which is able to differentiate at the beginning the two types of population would translate it?
I think in the stage of the high risk stage, so far it's not possible to differentiate whether a person will develop a schizophrenic psychosis or an affective psychosis. But the proposal by Pat that is suggested to broaden the high risk status is, of course, very helpful.
Okay. All right, so, thank you very much again for your presentation.